Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the lecture number 19 and we will discuss about two different microscopic methods. The first one is the optical tweezers and the second one is the traction force microscopy. So let us start with the optical tweezers. So the name tweezers brings this kind of an image to our mind in which it's a forceps which is known as a tweezer, and using this forceps you can hold a tiny object and you can either pull or push that object. You can put some level of force on the object using these tweezers, right? So similarly, the optical tweezers are laser based micro manipulators. So due to some physical properties imparted by the lasers, it can hold the object, the small objects, and using this laser beam you can actually uh, apply certain kind of manipulations to the trapped object. You can pull the object, you can push the object, or you can measure the forces that are being applied on the object. So let us understand what are optical tweezers. So optical tweezers use tightly focused light beam, that is generally the infrared laser, to hold and manipulate microscopically small objects. Okay, so microscope objective lens, generally of high numerical aperture, is used for the tight focusing of the IR laser beam, and this region becomes an optical trap that can hold small objects in 3 dimensions. Okay, so here you can see, so this is your glass cover slip and this is your laser beam. Okay, the IR laser beam, the infrared laser beam. So this laser beam acts as a trap. Okay, and it can hold the small object such as this bead. Okay, now this biomolecules like DNA or protein, they are actually very small. They are in the range of 1 to 10 nanometer in diameter. Okay, so directly you cannot hold the DNA or protein using this kind of laser beam, but you can actually hold the special kind of beads made up of dielectric material using this laser beam. So how you can use it in biology? So you can actually coat this dielectric bead with the biomolecules, maybe DNA, protein, RNA, and then using the laser you can actually pull or push this bead, or you can measure the force applied on the bead because of the conjugated biomolecules like the DNA or RNA or protein. So in this experiment you see DNA is one end of the DNA is tethered to the glass cover slip using some chemistry. Okay, we'll discuss it later. It's actually they have used this deoxygenin and anti-deoxygenin chemistry to fix one end of the DNA to the cover slip, and the other end of the cover slip, other end of the DNA, there is an enzyme known as RNA polymerase, okay, which is actually transcribing this DNA and producing this RNA. Okay, and this RNA polymerase is conjugated to this bead using this streptavidin chemistry, biotin streptavidin chemistry. Okay, so this RNA polymerase is conjugated with the bead, and the other end of the DNA is conjugated to the glass cover slip using this deoxygenin and anti-deoxygenin chemistry. Now using this laser beam, you can actually pull or push the bead, or you can measure the forces exerted by the RNA polymerase on this bead. So now this RNA polymerase is doing transcription on this DNA. Okay, so it will it has to move from this place. So it will apply certain level of force on the bead, and it will move the bead out of the central axis of this optical trap. Okay, so more it tries to go, now this optical trap works like a spring. Okay, so more this bead tries to go away from the center of the beam, okay, it will try to pull it. Okay, so bead will be attracted towards the center of the optical trap. So optical tweezers have been used with great success in the field of single molecule biophysics. For example, they have helped researchers unravel the complex elasticity and folding dynamics of DNA, RNA, proteins, especially the motor protein dynamics, and other long chain polymers. And these optical tweezers can also accurately measure the sub-piconewton forces exerted on the trapped object and the small displacements that the small biomolecules take place. Okay. So let us understand the physics behind the optical trap, that how this optical trap works. So dielectric particles are attracted along the gradient to the region of strongest electric field, which is the center of the beam. Okay, so this is the laser beam of the IR laser. Okay, and the strongest electric field is present at the center of the beam, like here. Okay, and this bead is made up of the dielectric material, and it is attracted along the gradient, along the gradient and to the region of the strongest electric field. So this is the center of the beam is this region of the strongest electric field. So this bead is attracted towards the center of the laser beam. The force applied on the object depends linearly on its displacement from the trap center, just as with a simple spring system. So what it tells, that the force applied on the bead depends on how much the displacement of this bead from the center of the laser beam. So let's say ideally this bead can be placed here at the center of the beam, and then there won't be any forces exerted on the beam because it is at the at the strongest electric field. But if you try to move this bead away from the center of the beam, then the force will be applied on the bead to pull it towards the center. Okay, so by Newton's third law, an equal and opposite force must act on the object, pushing it towards the center of the trap. So more you try to pull this bead away from the center of the trap, more force will be applied, and this bead will try to move back to the center of the this laser beam. So this is how the optical trap works. We will even look into the details of it, that how this light can apply force on the dielectric material. So the basic principle behind optical tweezers is the momentum transfer associated with bending of the light. Okay, so just understand and try to understand this figure to understand this paragraph. Okay, so here your laser light is here and your light beam is going this way. Okay, so what it's saying, the basic principle behind optical tweezer is the momentum transfer associated with the bending of the light. So this is a laser beam. Now when you put any object, let's say here a bead in the laser beam, then what happens, this bead will create some kind of a reflection or refraction of the light, and that will result in a change of the momentum of the light. Okay, because you are putting a bead in the light path, so it will create reflection and refraction. It will change the momentum of this laser light. So if an object bends the light and changing its momentum, as per the conservation of momentum principle, it requires that the object must undergo an equal and opposite momentum change, and this gives rise to a force acting on the object. Okay, so if this object changes the momentum, so as per the law of conservation of momentum, the object must undergo an equal and opposite momentum change. Okay, so this will create a force on this bead, and it will bring the bead back to the central position of this laser beam. So in optical tweezers setup, the incoming light comes from a laser, and basically the light at the center of the beam is brighter than the light at the edges. Okay, so the center of the beam contains the highest electric field. Okay, and that's why the light at the center is the brightest compared to the edges of the beam. When this light interacts with a bead, the light rays are bent according to the laws of reflection and refraction, and the sum of the forces from all such rays can be split into two components: the scattering force pointing in the direction of the incident light, and the gradient force arising from the gradient of the Gaussian intensity profiles and pointing in the xy plane towards the center of the beam. Okay, so when you put this bead in the light path or in the laser light path, the two forces will be applied on the bead. One is this scattering force and the other one is the gradient force. The gradient force is the restoring force that pulls the bead into the center. So this gradient force is the restoring force. It will always try to pull this bead towards the center of the light beam. If the contribution to the scattering force of the refracted rays is larger than that of the reflected rays, then a restoring force is also created along the z-axis, yielding a stable trap, and the image of the bead can be projected onto a quadrant photodiode to measure the nanometer scale displacement. So during this process, there is another laser and another optics. Okay, they actually uh, keep track of the position of the bead. So basically under the microscope you can see this bead, okay, using the camera, and you can see that how this bead is trapped in the laser, and when some force is applied, how this bead comes back to its original position. So when the bead is displaced from the center of the trap, what force does it feel? The restoring force of the optical trap works like an optical spring. The force is proportional to the displacement out of the trap, and in practice, the bead is constantly moving with Brownian motion, but whenever it leaves the center of the optical trap, the restoring force pulls it back to the center. So what it says, that this bead generally shows the Brownian movement when it is perfectly trapped and it is at the center of the beam. Okay, but when it goes away from the center of the beam, then this gradient force pulls it back to the center of the beam. And if some external object like a molecular motor were to pull the bead away from the center of the trap, a restoring force would be imparted to the bead and thus to the motor. So let's suppose if you have conjugated this bead with some kind of a biomolecule, some protein, motor protein or DNA or anything. And if those biomolecules tries to pull this bead, okay, then uh, this gradient force will try to pull this bead back to the center of the laser beam, and that force will be imparted to that biomolecule, whatever it is, the protein or the DNA, whatever you have conjugated with that dye. So this is how the force from the bead is imparted to your biomolecule. So here is an example trace of a single kinesin motor taking 8 nanometer steps against a 5 piconewton force is shown here. Okay, so you see this is a trace of a single kinesin motor, and it is making the displacement of the bead by 8 nanometer, okay, under the uh, uh, 5 piconewton force. So you can trace that single bead and how much displacement it is making and how much force is applied, that you can measure using this kind of experiments. So here is the microscope setup for doing this kind of optical tweezer based experiments. So you can see there are two laser lights. One is the trapping laser, that is the infrared laser, and other one is a detection laser. So detection laser can be regular, just like the way you use in fluorescence microscope. You can either have the fluorescent beads, or you can even see the beads under the bright field imaging as well. Okay, so both these lasers are combined here, then they illuminate the sample, and the position of the bead is observed by this quadrant photodiode. So this is a detector to observe the position of the bead, but you see that the red color IR laser will not go to the quadrant photodiode, so you cannot see the laser trap here using this photodiode. Okay, you will only see the image of the bead. So why you need such kind of method to measure such kind of a piconewton forces and the nanometer scale displacements? Because in biology, the forces and the step size are so small. Generally, the forces applied are in the level of piconewton, and the step size of the biological molecules is generally in the nanometer. Here I will show you one YouTube video in which you can see how what is the step size of a kinesin and how much force it applies on the cargo. Okay, so you can see here, this is a motor protein which is walking on the microtubule and it is pulling this cargo. Okay, so it has to apply certain forces to pull this cargo, and these are the step size that is it is making on the microtubules. So these step size are very small, okay, generally in the range of several nanometers, as we just saw that it was 8 nanometer, and these pulling forces are also very small, generally it ranges in the piconewton. So this is how the motor protein transports the vesicles or the cargo along the microtubules. Okay, so the concept that I want to convey here is that the forces and the step size in biology are very small, and to measure or to quantify such kind of things, you need a very sensitive method such as this optical tweezers, by which you can measure such kind of tiny level forces and the step size. Again, this is another example. People use this kind of technologies to study how study the forces required to break the kinetochore microtubule attachments. So these are the chromosomes, and during cell division, chromosomes are pulled from two opposite ends by the microtubules for the segregation of the genome into two daughter cells, right? So if you ask the question that how much force is required to segregate these two chromosomes, so you can measure such kind of forces using this kind of technologies, in which you attach these microtubules to one end of the glass slide. Then using the optical trap, you apply certain forces on these chromosomes or the kinetochores or the centromeres, and then you see how much force is required to rupture these kinetochore microtubule attachments. So we will see this in next slide. So this is one experiment in which people have studied the nucleosome dynamics using optical trap. So how they did it? So they fixed one end of the DNA using the deoxygenating, anti-deoxygenating chemistry to the cover slip. And this is a nucleosomal DNA, and the other end of the nucleosomal DNA is tethered to the bead. Okay, and this bead can be actually controlled by the optical trap, the lasers. Okay, so the more the force this DNA applies on the bead, and bead comes out from the center of the optical trap, the optical trap will pull impart force on the bead, so that bead should come back to its original position at the center of the beam. Okay, so this is how they reveal the nucleosome dynamics by disrupting the individual nucleosomes. So if you keep pulling the DNA using the optical trap this by moving the cover slip this side, then it will open up by releasing one after the another nucleosome. Okay, so here you can see the force extension curve of a single chromatin fiber. The cover slip was moved with constant velocity, and the DNA release started gradually in the low force range. Then in the higher force range, greater than 15 piconewton, the remaining DNA release occurred abruptly, giving a sawtooth pattern composed of 7 disruption peaks, which indicated 7 disruptions of nucleosome core particles. The force extension curve of a naked DNA molecule had no disruption peak, shown by the red dotted line. So what they are showing, so they made this kind of arrangement, okay, using the optical trap, and then they kept applying forces. So in the low force range, they didn't get this kind of they mentioned that it is a sawtooth pattern. Okay, so when this nucleosome opens up, it gives this kind of abrupt reduction in the force. So when the first nucleosome was opened, it gives this kind of abrupt reduction in the force. Again, the second nucleosome was opened, then it gave this kind of abrupt reduction in the force. So such kind of 17 peaks were observed. That means this nucleosomal DNA contains 17 nucleosomes. Okay, and they also performed the controlled experiment in which it was a naked DNA without any nucleosome, and when they pulled that naked DNA, they didn't find this kind of sawtooth pattern. Okay, because there are no nucleosome, so it showed this kind of curve, but when there were nucleosomes present, it shows this kind of sawtooth pattern. Then let us see this experiment. So here, using these optical tweezers, they measured the force required for breaking this kinetochore microtubule interaction. So these are the chromosomes, and the center of the chromosome, which are known as the centromeres, they are connected to the microtubules with the help of this green colored protein complex, which is known as kinetochore. So if you want to measure these forces, that how much force is required to break this kinetochore microtubule interaction, So what they did, with the cover slip, they fixed the microtubule, and then, so you can see with the cover slip, they fixed the microtubule, and one end of the microtubule contains the kinetochore, and they fuse that kinetochore with the bead. And then using the laser trap, they kept on pulling this bead so that until it breaks this connection. Okay, so what happened, this is a preload condition, and then they kept on increasing the force. So 0.25 piconewton per second. So you can see they kept on increasing the force, increasing the force, increasing the force, and at this point they found a rupture. Okay, rupture means this connection was broken between the kinetochore and microtubule. This connection was broken, and that's why it's an abrupt uh, uh, release or reduction in the force was observed, that they called as the force required for rupturing this connection. So how much force is required to rupture this connection? The 8 piconewton force is required to rupture this connection. Okay, so this is how you can quantify the force required to rupture this connection, or in the previous experiment you can measure that how many nucleosomes are present on the DNA. Yeah, so these are two major applications of this optical tweezers, that you can measure the biological forces and you can measure the step size of the biological processes. So let us move on to the second type of microscopy technique, which is known as traction force microscopy. So as you know, cells sense and respond to the mechanical properties of their environment. Okay, so the substrate stiffness can define the lineage commitment of the stem cells. And you can see there are different tissues mentioned here, and different tissues have different kind of stiffness. And generally the stiffness is measured in the elasticity of microenvironment. Okay, so you see this blood or this is fluid, then brain, muscle and collagen bones, all of them have a different stiffness. Okay, or the this is the elasticity of the microenvironment. And so how do the cells sense the stiffness of the substrate? So you can see, if you put the cells on the proper substrate only, then only it can grow and multiply. Okay, if the substrate stiffness is not correct, then cells may not grow. So how do the cells sense the stiffness of the substrate, and how you can measure that this is the stiffness is required for this particular cells? So what will happen if you put your cell on the deformable substrate? Okay, so cells exert mechanical forces on their substrate. So you can see, suppose if this is a silicon substrate and if you put a cell here, then cell will apply certain kind of forces on the substrate. So you can see this kind of wrinkles in the substrate. Okay, so can we measure the force this way, that how much force this cell is applying on its substrate where it is growing? Okay, so the traction force microscopy is required for the quantification of the cell and extracellular matrix forces. Okay, so measuring the forces on deformable and wrinkle-free substrates. So how this is done? So this is soft substrate, and on which you will put your cell. And these these small spots, what are these and what are they for? Okay, so those are known as the so this is a deformable substrate. Okay, so how it works, you see, if you see the SEM image, this substrate looks like a small pillars. Okay, and these small pillars are bendable, means they can bend. Okay, so when you put the cell, and if cell applies some force, these pillars will move like this. Okay, just like you must have seen this kind of balls, right? So they are made up of some flexible spikes. Okay, so these pillars are like this. So if cell applies some force on it, then these pillars will bend like this. And from the microscopic image and using the computational image processing, we can calculate that how much bending of the pillar has taken place, and from that we can calculate the amount of force that the cells has applied on the pillar. So here you can see the small pillars you are seeing, that is the substrate and on which the cell is there. Now different area of the cell imparts different forces on different pillars. Okay, so you can see some of the pillars moves a lot like this, whereas some of the pillars shows very small migration because of the forces exerted by the cell. So this is how you can measure the forces. So these small spots or the pillars, they are known as fiducial markers. Okay, so this is the soft substrate, and then you adhere your cell like this, and then you do live imaging and image processing and mathematics to measure the force. Now the cell will grow on this substrate and it will apply certain level of forces on these fiducial markers. Okay, so these pillars will move either to the left or to the right depending on the force applied, and that migration will be computed. So let me first show you that how it is computed. So before going into it, let me just explain the main assumption for the deformable substrates here. The substrate is purely elastic and it will not change over time, and there is no viscous component present, and the force deformation relationship is linear, so we can use the Hooke's law. Okay, so this is the conceptual framework. The deformable substrate is characterized by stiffness or Young modulus E bit displacement by delta L. So F is equal to E multiplied by delta L. Delta L is the displacement. Okay. So how to perform this experiment? Okay. So determine the substrate stiffness of your interest. Okay. So what is the force range of interest? Pascal or kilopascal? So as I showed that different tissues have different types of elasticity. Okay. So stiffness of your sample and its microenvironment. So from the literature you can get to know about it. For an example, if you want to see the fibroblast migration, you need the elasticity in the range of 5 to 10 kilopascal. If you want to see the immune cells, then you need the elasticity of 1 kilopascal. Okay. Then do you expect high or low forces during the biological process? As an example, cells exert more forces during migration than cytokinesis. So whether you are observing the cytokinesis or you are observing the migration of a cell, that you need to determine. And gel deformation must be less than 5%. The deformation deformation is the ratio of the difference between the deformed versus resting distance over the resting distance. Okay, so I will explain you this in the next slide. So the second question that you should ask is which elastic substrate to use, because there are different elastic substrates are available: polyacrylamide and polydimethyl siloxane. Okay, so PA or PDMS. So this polyacrylamide is used generally in the stiffness range of 100 pascal to 100 kilopascal, whereas this PDMS is used for this different stiffness. It has two types: Q-gel or Silgard. So this provides the stiffness range of 0.4 to 130 kilopascal, and this provides the stiffness range of 5 kilopascal to 1.72 megapascal. Now the advantage of this material is it is highly inert, so it is not going to interact with any of the chemicals or the media or your cell surface. Okay, where this PDMS, it is easier to modify adhesion geometry, and the refractive index is quite matching with the turf microscopy. Okay, and the disadvantage of this polyacrylamide is not compatible with turf because the refractive index is 1.33. So see, most of the time this kind of assays need the turf illumination. Okay, and for turf illumination the refractive index matters a lot, and that's why this polyacrylamide is not a turf compatible material, and this PDMS is hard to passivate. Okay, now the third step is the microscopy. So live imaging compatible microscope is required, and for PA gel you need confocal or epifluorescence microscope. For Q gel you can use turf mode. The objective, generally you need long working distance and high numerical aperture objectives. The 60x water immersion is optimum, or 60x turf if you are using a QGel, and 40x oil immersion objective can be used. And not compatible with DIC or phase contrast. So remove the DIC prism, and it cannot be used with the transmitted light. Only under the fluorescence mode you can use this. Now here you can see some representative turf images of the U2OS cell on the fibronectin coated 3 kilopascal Q gel. Okay, so these are the 200 nanometer beads, and this is the transmitted light image. Then this is the pexilin, the cell ECM adhesions you can see here, and the map of traction stress magnitude. Okay, and how it is measured, that you can see in the next slide. So for measuring the displacement, okay, you need two images. The zero force image, that is a gel alone, no active bit displacement, and the second image is for the gel plus sample. So here the active bit displacement you can measure. Okay, so the classical method is, this first step is acquire image of the gel plus sample and detach the sample and take a second image of the same position while avoiding any XY drift. The alternate method is acquire image of the gel alone, drop a cell, and acquire image after spreading. Okay, so there are two ways. Either you first take the image of the gel with sample and then remove the cells and then take the another image, or first you take the image of the gel only and then you put a drop of cell and then you take another image after spreading. So how it looks like. So see, this is a zero force gel image. Okay, so whatever the spots that you are seeing, those are the positions of those fiducial marker, the pillars. Okay, then you put the cell here. So the cells will displace the positions of the fiducial markers. Okay, so these pillars will move either to the left or the right or the up or the down. Okay, and that image will acquire in another color, maybe red color. No, sorry, the red color is given to the gel alone and the green color is gel plus cell. And then you see the merged image. Now here in the merged image, you see some of the fiducial markers are yellow in color. That means it's a superimposed image of red plus green. So yellow means there is no displacement. But wherever you see either the red or the green, that means it shows the displacement of those pillars. Okay, this is known as the bit displacement, and that is actually computed. So calculate the deformation field of the gel from plus and minus sample bead images. So this is a turf images of 40 nanometer beads in 3 kilopascal Q gel alone plus cell. So the red image represents the gel alone and the green image represents the gel plus cells. And then particle tracking velocimetry is performed using computationally, and then you can see that how those pillars have moved. So see, the pillars in this area moved in this direction, whereas the pillars in this area moved in this direction. So this is how the cell imparted forces on that soft substrate or the soft pillars. So this is how you can measure the traction forces applied by this cell. And using mathematics with the Fourier transformation, you can understand the traction force magnitude. So from this kind of heat map, you can see these are the areas where there was active or very high traction force was applied by the cell, whereas this is the area where the traction force was not at all applied. Okay, so this kind of analysis you can do using traction force microscopy. So that's it for today, and our next lecture will be on spatial transcriptomics and proteomics. Thank you for attending this lecture.